step into the laboratory of a revolutionary. The Genomic Guardian explores the transformative career of Nobel laureate Carol Greider, a scientist whose rogue approach shattered conventional biological wisdom. This presentation dissects the discovery of telomerase, the essential enzyme that prevents our genetic blueprints from unraveling during replication. By traversing the biological hierarchy, from the intricate dance of molecules to the vast complexity of entire systems, we witness how Greider's work serves as a foundational map for understanding aging and disease. It is a deep dive into the systemic impact of a researcher who didn't just study life, she identified the guardian protecting its very essence. Behold the cellular clock's greatest vulnerability, the mystery of the fraying ends. Every time a cell divides, it faces the end replication problem, a slow, inevitable unraveling of our genetic blueprints. Without a safeguard, this progressive shortening leads to genetic instability and cellular death. But nature has a solution. On the right, we encounter the telomere, biology's version of a shoelace aglet. These repetitive DNA sequences act as protective caps, shielding our chromosomes from degradation and fusion. In the early 1980s, science knew these aglets existed, but how cells actually built and maintained them remained a provocative enigma. This slide sets the stage for a discovery that would fundamentally change our understanding of aging and immortality. Witness a path of resistance, the remarkable journey of Carol Greider before she became a Nobel laureate. This timeline reveals a career forged not through standard metrics, but through sheer resilience. From early struggles with dyslexia and personal loss to being rejected by 11 graduate schools due to rigid test scores, Greider's story is one of bypassing systemic filters. Her unique cognitive approach, a lateral way of thinking born from necessity, allowed her to see what others missed. By 1984, she crossed the threshold into Elizabeth Blackburn's lab at UC Berkeley, ready to hunt for the mystery enzyme of life. It's a testament to the idea that brilliance often lies just beyond the boundaries of convention. It was Christmas Day, 1984, in a quiet lab at Berkeley. While the world celebrated, 23-year-old Carol Greider pulled a film that would change biology forever. After 9 grueling months of failed experiments, a glowing pattern emerged, the ladder in the darkroom. Using the pond-dwelling organism Tetrahymena, Greider identified a distinct, 6-base repeating sequence. Each rung on that ladder was the unmistakable signature of telomerase, the elusive enzyme capable of rebuilding the fraying ends of chromosomes. This single image provided the definitive proof that cells possess a mechanism for genetic maintenance. Greider's holiday solitude yielded a discovery that would eventually secure a Nobel Prize, unveiling the secret engine of cellular longevity. Step inside the molecular factory, where protein and RNA collaborate in a masterwork of biological engineering. Through ingenious experimentation, Carol Greider revealed that telomerase is no ordinary protein, it is a ribonucleoprotein. This sophisticated molecular machine carries its own internal RNA blueprint, acting as a built-in template to print protective DNA repeats directly onto chromosome ends. By using RNase to prove that RNA was the essential guide, Greider exposed the enzyme's secret sauce. It's a self-contained construction crew, the protein builds, while the RNA directs the design. This synergy doesn't just maintain DNA, it redefines our understanding of how life's most critical machines are built and operated. We now transition from pond dwellers to the grand scale of human biology. At just 27, Greider collaborated with Calvin Harley to prove that our telomeres act as a mitotic clock. Each cell division ticks closer to the end as these protective caps inevitably shorten. This slide illustrates the two fates of a cell. In Path A, telomeres run out, leading to natural aging and senescence. But Path B reveals a more sinister phenomenon, cancer. By hijacking telomerase, cancer cells restore their caps and achieve a terrifying immortality, dividing without limit. This pivotal work bridged the gap between molecular mechanics and systemic human health, uncovering the profound link between the length of our telomeres and the boundaries of life itself. Behold the telomere equilibrium matrix, the ultimate scorecard of cellular destiny. This slide illustrates that telomerase is neither a simple fountain of youth nor a cancer engine, it is a master regulator. In normal somatic cells, its absence triggers the ticking clock of aging. In cancer, its hijacking creates a rogue, immortal threat. Yet, in our stem cells, it behaves with surgical precision to ensure healthy renewal. It is the definitive biological tightrope. A delicate mechanism that sits precisely on the border between the grace of regeneration and the chaos of lethal disease. In this matrix, we find the chemical balance that defines life itself. We conclude with the Goldilocks balance, where the true stakes of Greider's research are laid bare. Using telomerase null mice at Johns Hopkins, her team discovered that organismal health exists only in a precarious equilibrium. To the left, we see the cost of too short telomeres, cellular senescence, and devastating syndromes like bone marrow failure. To the right, the danger of too long telomeres, the removal of cellular failsafes, leading to unregulated growth and cancer. This slide reveals the Goldilocks nature of our biology. We are caught in a constant tug-of-war between aging and malignancy, proving that the genomic guardian doesn't just build, it balances the very scales of life and death. The journey culminates in 2009 with the ultimate recognition, the Nobel Prize in Physiology or Medicine. Yet, for Carol Greider, this gold medal carried a weight far beyond its physical 6 ounces. As one of only 23 women among over 700 science laureates at the time, her achievement was a profound statistical anomaly. This slide, The Weight of the Prize, highlights the systemic gravity of her success. For Greider, the award wasn't just a personal triumph. It was a mandate to advocate for equity. She transformed her global platform into a duty, ensuring that the path she forged through resistance would remain open for the next generation of rogue scientists ready to challenge the status quo. We conclude by looking beyond the cell to the environment that nurtures discovery. In diagnosing the system, Carol Greider applies her analytical rigor to the culture of science itself. Using the iceberg of harassment diagram, she illustrates a stark reality: while institutions focus on the visible tip of explicit violations, they remain blind to the massive, submerged foundation of gender harassment and bias. For Greider, this submerged culture is what truly drives women out of STEM. By famously displaying this iceberg, she signaled that her duty as a genomic guardian included protecting the scientists themselves, ensuring that the future of research is defined by talent and equity, rather than the weight of a toxic system. In this slide, we see Carol Greider applying her analytical brilliance to the very structure of the scientific community. This slide, Restructuring the STEM Workforce, outlines a bold vision for reform presented in her 2019 science paper. Greider challenges the traditional academic model by proposing that social misconduct be treated with the same severity as data fabrication. She calls for radical transparency in salaries to eliminate gender pay gaps and a shift in incentives. Prioritizing mentorship and inclusivity over raw publication metrics. By treating systemic bias as a scientific failure, Greider seeks to engineer a healthier, more equitable environment. It is her final blueprint for a system where the pursuit of truth is supported by the integrity of the institution itself. In a notable stroke of rebellion, we witness the rogue biologist and the preprint revolution. Carol Greider's spirit of defiance extended beyond the microscope and into the gate-kept halls of scientific publishing. By 2016, she grew tired of discovery being locked behind paywalls and years of hidden peer review. She chose a different path, the Greider approach. By uploading her lab's research directly to the preprint server bioarchive, she demanded immediate, global, and transparent access. This small act of information age defiance helped ignite a global shift toward open science. It serves as a reminder that for a true genomic guardian, protecting knowledge means ensuring it is free, fast, and available to the entire world. This slide examines the power of the unconventional, where the disparate threads of Carol Greider's life converge. This Venn diagram illustrates a profound biological and social paradox. The very traits the scientific establishment often seeks to filter out are the exact catalysts required for paradigm-shifting discovery. By intersecting neurodivergent thinking, curiosity-driven basic science, and a commitment to systemic reform, we find the unconventional mind. Greider's dyslexia wasn't a deficit, but an engine for lateral thinking. her nose-following curiosity solved medical mysteries, and her defiance of the status quo now reshapes the STEM landscape. This is the ultimate lesson: to guard the integrity of our genome, we must first empower the rogue minds capable of seeing its secrets. In our final chapter, we witness a return to origins. In 2020, Carol Greider traded the burden of high-level administration for the purity of the lab bench at UC Santa Cruz. This is the unfinished blueprint, a testament to the idea that science is never truly complete. Greider views research not as a static destination, but as endless play, a puzzle where each solved mystery reveals 3 new questions. By simply following her nose, she transitioned from a curiosity about pond dwellers to the front lines of cancer research. It is a powerful closing reminder: the most significant breakthroughs arrive when we maintain the freedom to explore the unknown, ensuring the blueprint of life remains a living, breathing work in progress. We conclude our journey through the life and legacy of Carol Greider, the genomic guardian. From the microscopic mechanics of telomerase to the systemic challenges of scientific culture, her story is one of profound, multi-level impact. We have witnessed how her unconventional approach unlocked the secrets of cellular longevity and how she leveraged that global platform to advocate for a more equitable future in STEM. As the ladder of discovery continues to climb, we are left to consider the delicate balance between aging and immortality, and the courage required to challenge the status quo. Now, we open the floor to your inquiries, inviting you to explore the intersections of biology, resilience, and reform that define this remarkable scientific odyssey.